everything that we have talked about has been protein synthesis in eukaryotes. That's you. And you, 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 and frogs, and whales, and mice, and earthworms, and ladybugs, and cockroaches, and these plants over here, and this plant here. Eukaryotes. Those are animals, plants, fungi, some other little things called protists. We don't talk much about those. Those are eukaryotes. That's you. And when we talked about eukaryotes, we were talking about a nucleus, and we didn't talk about all the other organelles in the cell, but there's all kinds of stuff. What we've been wanting to get to, of course, is our much simpler little friends, the bacteria. Because that's who we're interested in, that's who we're talking about. So we've been talking about protein synthesis in bacteria, and we're going to take it back on track. You don't have to copy all this down. So this is just kind of a review. These are things you know about bacteria, prokaryotes. They're simple. They have circular DNA. They have some ribosomes, a cell wall. Some of them have flagella. Some of them have cilia. We know this. You don't need to write this down. We got some pictures of bacteria. The question that I've got, and I mean really, I guess we've kind of done this in backwards order, because the question I usually ask here is, do bacteria perform protein synthesis? We've been talking about it in eukaryotes. Obviously, you have all just read a bunch of articles that talk about protein synthesis in bacteria. You know they have to do this. If you didn't know that, I would say, well, do they have ribosomes?
Yes.
Yes. Do ribosomes make proteins? Yes. Do bacteria have proteins in their cells? Yes. Where do you think those came from? Protein synthesis. So we know that they're manufacturing proteins in the cell. What we want to know is how we can attack that process and not kill us in the process. One of you had a really good question is, how long could, if attacking protein synthesis kills bacteria, could attacking protein synthesis in a human kill the human? Yeah, it could. If your body's unable to make proteins, you die. Actually, even if your body makes proteins that aren't quite the right shape, have you heard of a condition called hemophilia? Hemophilia is a blood clotting disorder. People with hemophilia's blood either doesn't clot at all or doesn't clot very effectively. That means a simple paper cut can be deadly. Yeah. That's terrifying. That is the result of a single misshapen protein. A one gene mutation, the gene is changed, it doesn't make the protein with the right sequence, so the shape is wrong, so all the blood clotting factors that follow from that one protein don't work correctly, and those are people who could bleed to death from a paper cut. That's pretty crazy. That is crazy and terrifying. So, yeah, we don't want to attack our own protein synthesis. We need it. Don't break that. You're going to need it. But obviously we're attacking bacterias. So here's what I want you to think about. And again, you don't have to write all this down, but I'm going to have you come up with an answer at your table. Let me move this big chair out of the way. Steps of protein synthesis in a eukaryote. DNA unzips, we make an mRNA copy of a section of it, a gene. mRNA leaves the nucleus, goes out to the ribosome. Transport RNA starts bringing the amino acids over to the ribosome. The ribosome starts linking the amino acids into a chain, and when the protein's done, it stops. Sound reasonable? Sound like what we've discussed?
Yeah.
Okay, nobody argues with any of this. You shouldn't, because this is actually correct. No. You got no arguments with that. Good. So the question I have for you is, which one of those is not possible? Talk at your table.
I don't remember this stuff.
Okay. Which one of these steps is not possible in a bacteria? Talk at your table. Okay, let's actually look at a quick... Actually, I'm going to link this video clip on Classroom, and you're going to be responsible for watching it. It's like a 3-minute video on your own, because here's what I want you to realize. And this is, again, you don't have to write this all down.
Holy, oh my.
Yeah, you don't have to write it all down. I'm going to put this up on Classroom so you can see it. We got a list of things that only happen in eukaryotes. You just put your head up, though. Plants, oh, okay. Plants, animals, fungus, mushrooms, slime mold, ducks, blue whales. So in eukaryotes, mRNA gets out of the nucleus before protein construction can start. In you, every piece of mRNA codes for a single protein. Getting those proteins put together is a job that happens someplace else. So we sometimes have to link a couple proteins together to make another part of a cell. You are complicated. Don't ever forget that. Don't let anybody undersell you. You are a complicated and amazing multicellular organism. It takes 10 separate chemicals in you, or an earthworm, or a ladybug, or that plant over there, to start protein synthesis. So who here has ever run in a race? Okay, you hear the starter's pistol, you go, right? Imagine if you had to wait for 10 different signals. So first they count down, then they say go, then they wave a flag, then they run the starter's pistol, then they do something else, then a dove flies out across, I don't know what all happens. But 10 different things have to happen before you start running. That's essentially what goes on in your cell. There are 10 different chemicals that are required to start the process. That's pretty intense. The mRNA goes out of the nucleus, it goes to the ribosome, and it's just like cash register tape. It feeds through the ribosome. Your ribosome is pretty darn complicated, as a matter of fact. You could write that, I don't know. I have a complicated ribosome. Things I got going for me. I got a complicated ribosome. Bet you didn't know that. Well, so does that plant, so does an earthworm, so does a mouse, so does a dandelion. But that's okay. We're all complicated and multicellular organisms. In prokaryotes, I want to point out a couple big differences. So the big differences I want you to notice are this. There are only two starting